Chapter 13, Meiosis, Sexual Reproduction. In this chapter, you will learn how meiosis promotes genetic diversity and allows for the benefits of sex. We will talk about how does meiosis occurs, and we will compare this to what we've learned so far about mitosis. We'll talk about the importance of how meiosis promotes genetic variation, and it does it three ways. It does it through independent assortment, crossing over, and fertilization. What happens when things go wrong? We'll also talk about the benefits of meiosis through purifying selection hypothesis and the changing environment hypothesis. When we talk about sexual reproduction, what are we talking about? We are talking about sperm and egg, and they unite to form a fertilized egg or a zygote. So sperm and egg are called gametes. So gametes are sperm and egg. Now, the thing about meiosis is that it goes through a nuclear division and it cuts the chromosome number in half. So human beings, for example, have 46 chromosomes, but human sperm or human eggs only have 23. In other words, the chromosome number is cut in half. If we have a frog that has 20 chromosomes, then the sperm and the egg from that frog will only have 10 chromosomes. You can expect to have test questions that ask you the number of chromosomes in gametes. Every organism has a characteristic number of chromosomes. So we know that fruit fly has 8 chromosomes. When I took my genetics classes maybe 30, I hate to say this, over 30 years ago, all that was discussed in a genetics class was one fruit fly experiment after another, and this is how researchers began to understand meiosis. So there's two types of chromosomes. The two types are autosomes and sex chromosomes. Autosomes are non-sex chromosomes. And then you have your sex chromosomes. Two X's make a female, and an X and a Y make a male. So two X's a female, an X and a Y a male. This drawing sort of seems strange to me, but it reminds me of a new term that we're going to come and see in a little bit called homologous pairs. Like the long chromosome from a male will match with the long chromosome with a female. The short one from a male will match up with the short one from a female, and so on and so forth. And then these will be called homologous pairs. Homo meaning the same. Chromosomes of the same type are called homologous pairs. So let's think about this for a moment. You know that I'm an old lady, and but that still doesn't stop me from missing my parents. Like I miss my mom and I miss my father who passed away some time ago. And sometimes I'll go like this to myself, and it's not to calm myself down, but it's to remind me that every single cell in my body is half my mom and half my dad. So when people say your parents are always with you, they literally are always with you. I am one half of my mom and one half of my dad. And when we pair up the chromosomes, these are called homologous pairs. The long chromosome from dad matches with the long chromosome from mom. And the short one from dad matches with the short one from mom, and mid-size to mid-size. Now, remember from the previous chapters that we have genes on each of these chromosomes, and genes is enough genetic material to code for a protein or a trait. Now, variations of these traits are called an allele. You can see here that I have very thick, dark hair, and it is curly. Okay, so these are all genetic traits. Thick hair, frizzy hair, dark hair, all of these are variations of traits. So here's another example of alleles. So we're talking about eye color. So on one gene, there's a gene for red eyes, and on another chromosome is a gene for purple eyes. These are called alleles, variation of eye color. So red versus purple, variation of a trait. The next term I'd like to talk about is karyotype. A karyotype identifies the number and types of chromosomes present in a species, and this is done under lab conditions. So I remember when I was pregnant with my last child, I was 38, and 38 was considered to be a relatively mature mom. So they were scared that I might have a child with trisomy 21 or Down syndrome. So they did a karyotype doing amniocentesis. They mapped out the chromosomes. When they mapped out all of the chromosomes of my baby, then this is called a karyotype. When you see pairs, then we're talking about the diploid condition, 2N, one from mom, one from dad. If we have half the chromosome number, then we're talking about the haploid number, just N, one set of chromosomes, where diploid or di means two sets of chromosomes. Each species has a haploid number. So for example, humans, 2N equals 46, so N would be 23. 2N is the diploid number, N equals 23 is the haploid chromosome number, and 23 chromosomes are found in either sperm or human eggs. Now, there are organisms that have multiple sets of chromosomes. I'd like you to be aware of that. But for this chapter and for the test, you do not have to know about polyploidy. But usually in the plant world, we have multiple sets of chromosomes. So when we talk about diploid or 2N cells, we're talking one set comes from mom, maternal chromosomes, and one set comes from dad, paternal. So both of these together is 2N. And as I mentioned before, you can have multiple sets, like cotton has 6 sets of chromosomes, strawberries have 8 sets, and it's usually in the plant world, but let's stick to the animal world that has 2 sets, diploid. So here's 13.1 is a very important and long table. Please make sure that you know most of the terms and you know the examples. So here they talk about sex chromosomes, autosomes, unreplicated chromosomes, replicated chromosomes, sister chromatids, homologous chromosomes, non-sister chromatids. So here's a replicated chromosome from mom, and here's a replicated chromosome from dad. So when they cross over, we have non-sister chromatids touching each other. We have the term bivalent, but I'll be honest, I don't use that term very often, and I won't assess you on that term. But you will have to know haploid number, diploid number, ploidy, haploid, diploid, and polyploid. Okay, now let's talk about meiosis. So meiosis, remember, cells go through the cell cycle and they can either go through mitosis or meiosis. Now, when you go through meiosis, what are you forming? You're forming actually sperm and egg. This is gametogenesis. So I'm going to be honest with you. I have 3 children. They're all grown. They're all like around 30. Some are a few years younger than 30 and some are over 30. But when I had my children, my husband said, "Do you want to make a baby?" I said, "Yes." And he started going like this. He started coming to me and he said, okay, let's make a baby. And I did the same thing. I started going to him this way. So when I go like this, what am I talking about? I'm talking about what? New genetic variations. Why is that so important? So remember, when any organism, including humans, have sex, it's very time consuming and it takes a lot of energy. So why would organisms do something that's very time consuming and takes a lot of energy? I always say when you are ready to find a partner and mate and have sex, you have to be nice to that person, you have to shower, brush your teeth, you have to eat properly, you have to pretend that you're interested in what they're saying when they speak. So why spend all of that time getting ready for sexual reproduction? Because the purpose of sex is to have new and wonderful genetic combinations so that the next generation has a chance to survive in an ever-changing environment, has a chance to survive in spite of genetic mistakes that are made. And that is the purpose of having meiosis, or the formation of sperm and egg. So it says, just before meiosis begins, each chromosome in the diploid parent cell is replicated. So here we have one chromosome and then it becomes replicated. So you've gone through this before, and these replicated chromosomes, as long as they're attached in the centromere, they're called sister chromatids. So what So something that is new, it says meiosis consists of 2 cell divisions. So remember, in mitosis it was pay me anytime, boom, done. But in meiosis there's a meiosis 1 and meiosis 2. So what is the purpose of meiosis 1? The purpose of meiosis 1 is to separate homologous pairs. What is the purpose of meiosis 2? To separate sister chromatids. And this is so important. So let me repeat this. The purpose of meiosis 1 is to separate homologous pairs. Maybe another way of putting them, one from mom and one from dad, is once they cross over is to separate them. And this cuts the chromosome number in half. Then the purpose of meiosis 2 is to separate sister chromatids. So here you see meiosis 1 and 2. What else is new in this chapter is you notice now we have chromosomes in color. We have red and blue indicating those coming from mom and dad. So notice in meiosis 1 up at the top, how many chromosomes are there? Yeah, you're right. There's 2 chromosomes, 2 replicated chromosomes. And then at the end of meiosis 1, notice that the chromosome number is cut in half. And then notice in meiosis 2, sister chromatids are separated. And so we end up with 4 cells. And each of these cells will be genetically different. Meiosis consists of 2 cell divisions, meiosis 2 and meiosis 1. So let's take a look. If we compare it to mitosis, in mitosis we only have 1 cell division. So as in mitosis, chromosome movement is coordinated by microtubules. So we have a spindle fiber apparatus and we do have kinetochores. And these kinetochores will help guide the chromosomes, on microtubules to opposite poles. Okay, meiosis 1 is reduction division. So what does that mean? It means that one cell is divided into 2, and at the same time the chromosome number is cut in half. So 2 things are happening. And notice another way of saying this is gametogenesis, the formation of egg and sperm. Okay, so remember, at the end of meiosis, we're going to have 4 genetically different haploid daughter cells. So fertilization results in a diploid zygote. In other words, half the chromosome number and half the chromosome number makes a whole. So we restore it. Let's look at humans. 23 plus 23 makes 46. In figure 13.4, you can see that we have a female gamete and a male gamete. Those are haploid. And haploid plus haploid makes a fertilized egg, which is called a zygote. Meiosis 1 is reduction division. In other words, we start off with one cell and we end up with two. We start off with two sets of chromosomes, but we end up with one set. Now, what does this mean, like in everyday stuff? If you look at your features, like my daughter will say, Mom, why do I have such a big nose? Or why do I have a long face? Or why do I have such a thick brow? Well, it's because dear old mom has bits and pieces of her own family where there were thick brows, long faces, sort of succulent lips. That's how she got these things. And so my traits are mixed up with those of my husband's family's traits. And so this is this cycle of new genetic combinations. In figure 13.5, you see this mature dog right here, which is diploid, has two sets of chromosomes. But in the formation of egg and sperm, meiosis happens. One cell will form four cells, either four sperm cells or four egg cells. And each of these daughter cells will be genetically different. And two of these will combine to form a fertilized egg or a zygote. And notice that the chromosome number is then actually retrieved, or it becomes restored. It's the same. So this is the cycle of life. Okay, so what are the phases of meiosis 1? Pay me anytime. Pay me anytime. I'm not going to talk about early prophase or late prophase. I'm going to group them into one. Just prophase 1, metaphase 1, anaphase 1, and telophase 1. So what happens in prophase 1? Prophase 1 is very important and unique, and you should be prepared to receive many test questions where the answer is prophase 1. Why is it so important? Because not only does the nuclear envelope begin to break down and chromosomes condense, but homologous pairs begin to form. And this is something new. We never saw this in mitosis. This is something new. And this is when the homologous pairs come together, whether you see it this way or on top of each other like this is called synapsis. And so bits and pieces of information are going to be shared between both maternal and paternal chromosomes. So when they share and swap little bits and pieces of genetic information, this is done at joints as they pull apart called chiasmata. Chi is X, is the Greek X. So these X's are, that's why they're called crossing over. At these crossing over points, we see that bits and pieces of genetic material are being exchanged between maternal and paternal chromosomes. During metaphase, something else is happening, which is very important, and you can expect to get a lot of questions on this coming up in your next test. And this is that the chromosomes line up in two rows. So they're homologous pairs, right? So they line up in two rows. It can be like this, mom and then dad, or it could be dad and then mom. Each one is, it could be in either way for each pair, and this is called independent assortment. Independent assortment. And you can imagine how profound this is, because as the homologous pairs separate, then you can see some traits will go to some of the future eggs and sperm, and other traits will go to other eggs and sperm. And this is creating many more new genetic combinations, just like crossing over can create so many, infinitely many combinations. Then in anaphase 1, the homologous pairs separate and migrate to opposite poles. In telophase 1, homologs finish migrating to the poles, and then they separate, forming two cells, cytokinesis. So therefore, at the end of meiosis 1, homologous pairs have separated, two sister cells or daughter cells have formed, and each of these cells are genetically different. So here we see crossing over, and I love the pictures that are in your book in figure 13.6. First of all, I guess one of the first questions I would ask is, how many chromosomes are we starting with? I'm going to count them. If I count them, I see 1, 2, 3, 4, 5, and 6. There's 6 chromosomes and there's 3 pairs. Do you agree? Do you see that they're crossing over and bits and pieces of information are being exchanged? You can actually see the chiasmatoform in step 3, late prophase. Do you see the little X's forming? And then in step 4, we have metaphase 1, where we see that we have pairs lining up in the middle. What makes it unique is that we have 2 rows of chromosomes, indicating the homologous pairs. The red ones, let's say they're maternal, so we have independent assortment. They can line up independent of each and every other pair, and this is creating many more combinations. So notice by anaphase 1, they're separating. Homologous pairs are separating, and they're migrating to opposite poles. As mentioned, meiosis 1 results in daughter cells with only chromosomes of each homologous pairs. So the homologous pairs have separated. Homologous pairs have separated. Okay, so now cells are haploid at the end of meiosis 1. They have a random assortment of maternal and paternal chromosomes. So this is random or independent assortment. And you could be asked a question, when does random or independent assortment occurs? It occurs during metaphase 1. When does crossing over occur? Crossing over occurs during, yes, you're right, prophase 1. Okay, so now let's move on to meiosis 2. In meiosis 2, what is the purpose of meiosis 2? The purpose of meiosis 2 is to separate sister chromatids. Okay, so this is almost equivalent to actually mitosis. So you should say, pick my nose, I already understand this. So do we have 4 distinct phases? Yes, we do. Pay me anytime. Prophase, metaphase, anaphase, and telophase. In prophase 2, the spindle fiber apparatus forms again, and microtubules from each pole attach to kinetochores on opposite sides of each chromosome. During metaphase, they line up in the middle. During anaphase 2, sister chromatids separate, and the resulting daughter chromosomes begin moving towards opposite sides of the cell. When chromosomes arrive at opposite sides of each cell, a nuclear envelope begins to form, and then eventually each cell undergoes cytokinesis. So here we see the end of meiosis or meiosis 2. So notice meiosis 2 looks almost identical to mitosis because the purpose is the same. It's to separate sister chromatids. So let's recap. In meiosis 1, we separate homologous pairs. In meiosis 2, we separate sister chromatids. So at the end of meiosis 2, we end up with 4 haploid daughter cells, and every single one of these daughter cells are genetically different. One diploid cell with replicated chromosome gives rise to 4 haploid cells with unreplicated chromosomes. You can look at this video if you go back to the presentation posted in Blackboard under presentations. Okay, so what are some key differences between mitosis versus meiosis? Well, mitosis is for asexual reproduction. Meiosis is for sexual reproduction. Okay, so what are some key differences? There are homologous pairs, but they're only found in meiosis. There are no homologous pairs found in mitosis. The pairs separate during anaphase 1, cutting the chromosome number in half. Remember, in mitosis, the chromosome number always stays the same. You start with one chromosome number and you end with the same chromosome number. In meiosis, you start with one chromosome number and you end up with half the number. Mitosis produces two diploid daughter cells, and they're genetically identical. In meiosis, there's 4 cells that are produced, and each one is genetically different. In this drawing here, you can see the differences between mitosis and meiosis. So this gives you a visual representation of some of the differences. Table 13.2 is a wonderful table to compare mitosis and meiosis. I strongly encourage you to look at this table. Meiosis promotes genetic variation. So remember, in mitosis, every single cell is a clone of the parent cell. But in meiosis, every single gamete or every single sperm or egg that is formed is actually different. There's a shuffling of maternal and paternal chromosomes. And this has to do with crossing over during prophase 1 and independent or random assortment in metaphase 1. Now, the third source of variation is the last bullet on this slide. The third source of variation is fertilization. When I was doing the dance for new genetic combinations with my husband, when I had my 3 children, he never said, "Hey honey, I only want that particular egg." Nor did I say to him, "Hey honey, I only want that sperm. I don't want any of the other sperm." So in other words, fertilization was a very random event. The only time that I can see that fertilization is not a random event is when we use technology to fertilize an egg. Meiosis promotes genetic variation, and remember that's important. Like when you walk into a building or into a classroom or into a store, you don't expect to find a clone of yourself. In fact, everyone is so different. Everyone is expected to be different, and it happens so consistently that we just take it as a matter of fact of life. We don't even think about it anymore. But actually, that's what is one of the most important underlying principles of life itself, is genetic variation and how important it is to life. So sexual reproduction is a very important part of reproduction. Remember, asexual production produces clones. Sexual reproduction produces offspring with unique chromosome complements. What is independent assortment? So we've seen it as independent assortment or random assortment, and it happens during meiosis 1. More specifically, it happens during metaphase 1. And this is where the homologous pairs, the pairs line up, mom, dad, mom, mom, dad. They line up on top of each other during metaphase 1. So there's 2 rows of chromosomes. And this independent assortment is going to give rise to genetic diversity. So they give you an example of how important it is when we see the chromosomes line up in the middle. So in this example, they used two genetic diseases, sickle cell anemia and cystic fibrosis. I wouldn't wish either disease on any person, but depending on how the chromosomes line up, you could end up with no diseases, or you can end up having both cystic fibrosis and having sickle cell anemia. So the way the chromosomes line up will affect the traits that you inherit. What is the other source of genetic variation besides fertilization, besides independent assortment? Yeah, you're right. It's crossing over during prophase 1. So these new combination, those bits and pieces. So when you look at yourself and you look at your parents and your extended family, and you see that you're a combination of bits and pieces of your family, are you in fact a combination of bits and pieces of your family? And the truth is you literally are, because of crossing over during prophase 1. I remember that I had a teacher that I adored in 2nd grade, and she would tell us, I believe in God because the face of every little child that I've had as a teacher has been different. But I've always wanted to go back in time and say that really the reason is because of meiosis. It's because of crossing over, independent assortment, and because of fertilization. Each gamete is genetically unique, so when a given sperm and egg come together, it's random. And so this random joining also increases genetic variation. So this is the third source of variation. Okay, so let's talk about what happens when things go wrong. One of the most common errors when things go wrong is Down syndrome. So oftentimes you can have errors. That means that chromosomes are not dividing evenly during meiosis. In other words, we can say that they're sticky chromosomes. The more proper word is non-disjunction. So one of these errors is to have a sticky chromosome for the 21st pair, and it is called trisomy 21. So instead of having 2 chromosomes, there are actually 3. And this can happen in 1 in every 700 live births in the United States. So in this diagram here, you can see a karyotype has been done, but if you look at the 21st pair of chromosomes, you notice that there are 3 chromosomes instead of 2. This is Down syndrome, trisomy 21. The sticking of chromosomes is due to non-disjunction. In other words, chromosomes stick to each other and they don't divide evenly. So it can happen that both sister chromatids move to the same side, or homologous pair chromosomes, the pairs do not separate. They move to the same side, and this is called non-disjunction. So either gametes will have too many chromosomes or too few. So here you see an example where you can see non-disjunction occurs in step 2 in meiosis 1, where the homologous pairs, there's a pair that goes to one side. They stick to each other, non-disjunction. So 2 chromosomes have one too many, and two of the cells have one too few. Now, we can call this monosomy or aneuploid, but the important thing for me to convey to you is that non-disjunction does occur. So you can, I won't hold you responsible for monosomy or aneuploids, but you do have to know about non-disjunction. Why do mistakes occur? So it's sort of hard to visualize because I think oftentimes we want answers for everything, but sometimes these errors are just random errors. In other words, you can't put a connection to them. So it can happen during human egg development where um, primary oocytes uh, during during embryonic development do not. Some of the chromosomes stick to one another. So meiosis does not continue until ovulation, and so that's why oftentimes non-disjunction occurs in older eggs, because the end of meiosis occurs at the very end of the cycle of egg development. When I had my son, my last child, I was 38 years old. So what was the probability or the incidence of having a baby with Down syndrome? So if I look at the graph and I find my age on the x-axis, I see 38, and yes, you're right. It's 1 in 200 births is the incidence of having a baby with Down syndrome. And notice that it exponentially increases as a woman gets older. You can also watch this video on the presentation posted in Blackboard. Okay, let's recap. What are the benefits of meiosis? Well, we know that sexual reproduction takes a lot of time and energy, but why is it so important? Why is it beneficial? As it is, if you have asexual reproduction, then every cell can reproduce. With sexual reproduction, you cut that number way down. This is often referred to as the paradox of sex. So notice in asexual reproduction, a single cell can produce way more cells than two cells having to get together. And this shows you the comparison of offspring between those that are asexually reproduced versus those that are sexually reproduced. Yeah, so why have sex if you have a way less and it's time consuming and expensive? Well, this is called the paradox of sex. And asexual reproduction can yield twice as many offspring because every single parent can produce its own offspring. But the reason is that we have sex is because there must be some special kind of benefit. So one hypothesis for this benefit of having sex is called the purifying selection hypothesis. So what does that mean? That means, let's say there's a mutation. If there was a bad mutation and everyone was a clone, once you had a mutation, then everyone would be affected. And if it was so bad that it killed the organism, then that would wipe out the whole species. But when you have sexual reproduction, you have one chromosome that has the deleterious allele, but you also have the other side, the homologous pair. So what happens over time, there's a steady reduction without devastating a population. So if a deleterious or negative allele appears in a sexually reproducing individual, not all offspring will receive it, maybe at most half. The other idea for the benefit of sexual reproduction is the changing environment hypothesis. Offspring that are genetically identical to their parents are unlikely to thrive if the environment changes. And so in an ever-changing world of changing environments, having new genetic combination means that there's a higher probability that offspring will survive, especially if they're exposed to emerging pathogens. So asexual reproduced offspring will be equally susceptible to new strains of pathogens, whereas those offspring that are genetically different, some will have more resistance than others. In this study, they were studying worms that lived in ponds. In one pond, there were no pathogens attacking the worms. In another pond, there were pathogens that were attacking the worms. So if you look at the x-axis, you look at the number of generations, and the y-axis is the rate of outcrossing, or the rate of sexual reproduction. So notice in the pond where there were a lot of pathogens attacking the worms were reproducing sexually. But in the pond where there were no pathogens, then the worms produced asexually. So this gives this idea that new genetic combinations will provide ammunition against emerging and new pathogens. Studies support the changing environment hypothesis. Genetic variation promoted by sexual reproduction may be favored in certain environments, those environments that have pathogens. And this is the end of meiosis.